In this video, I'll be talking about point mutations. Here we have a DNA sequence, and we are told that it creates a protein that is 4 amino acids long. So that means that we need to find the ATG codon in the first or second strand. We must determine which one is the correct reading frame. So that means since there's a 4 amino acid protein that is created, that means that there are 5 codons in this reading frame, including the stop codon that codes for the stop transcription. So looking in the first strand, we see that the 5' and 3' directions are already given to us. And so the only ATG in that first strand is the very first 3 ATGs right there that is seen. And so the next the next codons that we see are TGG, ATT, ACT, and TGC, which will eventually actually be the incorrect reading frame because it does not end with a stop codon. Now in the second strand, we have an ATG that is going in the other direction, 5' to 3' still, but it's reading right to left instead of left to right. So we have ATG, GTC, AGC, AAG, and TAA, which will be the correct reading frame because it ends with a stop codon. This is just the codon dictionary that we'll be using to piece together the amino acid that's created. Here I'll be showing what the mRNA will be transcribed from the DNA. So I swapped the direction, so it's now reading 5' to 3' left to right correctly on the coding strand. And so you see it's ATG, GTC, AGC, AAG, and TAA. And the mRNA will just be the same thing, except all T's are going to be coded to U's. And so below that you see the amino acids, and we have the start with this methionine. After that, valine, serine, lysine, and then the stop codon. And this is the wild type mRNA, so it will have no effect on the organism. Here we have a missense point mutation, and it's a transversion because it is a mutation from a T to an A, which is a pyrimidine to a purine. And so it will make the valine turn into aspartate, and it will create a different amino acid, which will most likely lead to a non-functional protein. Next, we have a nonsense point mutation, and it's also a transversion because it's an A to a T, a purine to a pyrimidine, and the mRNA will now be UAG, which codes for a stop codon, so it will have a short amino acid sequence, and it will lead to a non-functional protein. Here we have a silent point mutation, and it's a transition because it's from a T to a C, which is a pyrimidine to a pyrimidine, and the mRNA will now be GUU, but it still codes for valine, so it's going to be a normal protein sequence because the amino acid sequence is the same. Lastly, we have a read-through point mutation, and it's a transversion because it's from a to a C, which is a purine to a pyrimidine. And so now we will have an amino acid that is lengthened because the stop codon has turned into a different amino acid, which in this case is a tyrosine, and it will make an extended and non-functional protein.